So good morning to all of you. This time we are going to discuss on the design of fermenter. So design of fermenter, basically fermenter required the lots of sensor, lots of contamination free aseptic conditions. So if you see the figure, that is acid, they have required acid base control, anti-foam control, exit glass flow, fresh media, feed, level sensor, pH sensor, dissolved O2 sensor, agitator, sparger, exit level. So lots of sensors, lots of control points, input, output are required in the fermenters. So fermentation process, as you know, there is a stock culture. We have the stock culture, then we give we use this stock culture as a working culture to the seed fermenter. We use production fermenter. We put this stock culture or working culture in this production fermenter. Then we have the biomass production, cell separation, and then purification, characterization. All these in this fermentation process take place. So fermenter was first described by uh, Carl Weizmann in 1914 to 1914 to 1980 during the First World War. So he developed a process for the production of acetone by liquid fermentation using clostridium. That was the first he developed the process. Then in the large scale, the first uh, aerobic fermenters were used in Central Europe during 1930s for the production of compressed yeast. So the fermentation process is not very new, as already we discussed in the history. And this fermentation process required aseptic condition. It means fermenter design should be aseptic condition. So aseptic condition that required good aeration required, agitation should be there, essential and important factor for the production of good product. Basic function of fermentation is long-term operation with aseptic condition, adequate aeration and agitation, low power consumption, control temperature, control pH, sampling facilities, not excessive evaporation to lose the watery content, minimum use of labor operation, suitable range of operations, ensure smooth internal spaces, similar geometry to both smaller and large scales, and most important, cheapest material we can use, and adequate service provision should be there. So fermenter should be constructed in such a way that it may have provision of the several activities like sterilization, temperature control, pH control, foam control, aeration, agitation, sampling point, inoculation point, drainage system, harvesting product, and cleaning. So various component is required during the process. Various parts are required for the fermentation fermenter. There's a top plate, clamp, seal, vessels, drive motor, drive shaft, marine impeller, baffle, sparger, exit go, gas cooler, inoculation needle. Several parts are there. Several monitoring and control parts of fermenters are used: foam probe, pH electrode, temperature control, oxygen sensor, heater, pad, cold finger, rotometer, pressure valve, air pump, parasitic pump. So these are the control system of the fermenter. So if we discuss on the basic function of the fermenter, it should provide a control environment, permit aseptic condition, provide adequate mixing, aeration, optimum growth production without damaging the microorganisms. Power consumption should be minimum. It provides easy and dependable temperature control. Facility for sampling should be provided. Evaporation loss should be low possible. It should require a minimum of labor. It should be suitable for range of fermentation process, but this range may be often restricted by the contaminant regulations. So these all the why we require the aseptic for the basic function of the fermentation. We require these things for proper functioning of the fermenter. So if we talk about the fermenter design, a bioreactor is a device in which a suitable of low value is utilized by living cells or enzyme to generate a product of higher value. So we are exploring the microorganisms for developing the enzymes, antibiotics, or desired product. So bioproducts are extensively used for food processing, fermentation, waste treatment, and many more things. So based on this fermentation, there may be a bioreactor can be grouped into two broad classes: those based on living cells and those implying enzymes. So maybe they may be a aerobic, anaerobic, solid waste, immobilized cells bioreactor based in terms required for the process requirements. Theoretically explanation, usually bioreactors should be provided for the following: agitation and aeration. So these two things, agitation and aeration, are the basic and important uh, technical point through which we can get higher or desired product. So agitation, the medium must be suitably stirred to keep the cell in suspension and to make a culture homogeneous. Proper homogeneous, proper equilibrium should be there. Homogeneous environment should be there. It becomes increasingly difficult with the scaling up. So if there are homogeneous, non-homogeneous condition is available, then it is difficult to scale up. Various type of stirrer are arranged in the bioreactors and the fermenters, like magnetic stirrer, flat blade turbine impeller, marine impeller, and pneumatic energy, air-led fermenter, and those using hydraulic energy, for example, medium perfusion. Aeration may be achieved by medium perfusion, in which medium is continuously attacked, continuously taken from culture vessels, pass through an oxygenation chamber and return to the culture. So perfusion is used with the glass beads and more particularly with the micro carrier systems. The following components of the fermenters are required for aeration and agitations, like impeller, like stir glands and bearing, baffles, and the aeration system disperser. If we discuss on these aeration component, not process component, we can say first we take agitator, impeller. Agitator achieve the following objectives: bulk fluid, gas phase mixing, air dispersion, oxygen transfer, heat transfer, suspension of solid particles, maintenance of uniform environment throughout the vessels. So the main objective of the agitators or impeller is to mixing, air dispersion, oxygen transfer, heat transfer, maintains the uniform environment throughout the vessels. These objectives are achieved by the suitable combination of the most appropriate agitators, air disperser, and buffels, and the best position for nutrients feeds, acids, air, alkali, pH control, or antifoam and additions. So agitators are of several different types. Disk turbines are there, vent disks are there, open turbines of variable pitch are there, propellers are there. So different types of agitators are available in the market also. So you can use these agitators. So impellers are an agitation device. They are mounted on the shaft to introduce the fermenter throughout the lid. These impellers are blades or attached to motors on lid. If you talk about this, there are different types of propellers, turbine, disk turbine, wind disk turbine, variable pitch open. So different types of agitators are available. So in the fermenter, there may be a single multi-blade impellers, or there are three or more multi-blade impellers are available. So impellers are all helps in breaking foam bubbles in the head space of fermenter. This foam formed during the fermentation process can cause contamination problem, and these problems are avoided by the use of. So they make the homogeneous mixed conditions environment, mixing the organisms, uh, breaking the bubbles, foams to make equal environments. Buffers. Buffers are metal strips roughly 1/10 of the vessel's diameter and attached readily to the fermenter wall. They are normally used in fermenter having agitators to prevent vortex formation and to improve aeration efficiency. So if you see this, you can find the buffers here. So agitators, and then here is the buffers which are mounted on the wall of the fermenter. The important function of buffer is to break the vortex formed during the agitations process by impeller. If this vortex is not broken, the fermentation media may spill out of the fermenter, and this may result in contamination of the nutrients, contamination of the media, which make a huge problem. So it is important to break the vortex formed by using a barrier. Buffer act as a barrier which break the vortex. You can say this: buffers act as a barrier which break the vortex. This would lead to a scoring action around the and behind the buffer, which would minimize microbial growth on the buffer and the fermenter wall. If we talk about the aeration system, so there is a device used to introduce air into the fermenter broth is called sparger. Spargers are the following three basic types: porous, orifice, and nozzles. So this may provide the aeration in the fermenter through which the sterile air is introduced in the fermentation tank. Porous aspergers may be made of sintered glass, ceramics, or a metal. Aspergers are located on the bottom of the fermentation tank. Glass wool file filters are used in asperger for sterilization of air and other gases. The air released in the form of tiny air bubbles. These air bubbles help in the mixing of media. So aeration system is important. We are giving the sterile air in the form of bubbles which help in the mixing of media. So if you see the diagram of the fermenter, so you can find the aseptic inoculation of pipe, stir soft seal, working level of broth, buffer, sampling point, impeller, sterile air line, air sparger, drain point. So this is the uh, diagram of the basic design of the fermentation. So there is a sampling point also, where sampling point is used to time withdrawal of sample to monitor fermentation process and quality control. What about the we have the pH control. So we have to control the pH of the media. Device check the pH of media at a specific interval of time and adjust the pH to its optimum level of addition of acids or alkali. So maintaining the pH is required, is important for the uh, growth of microorganism to obtain a desired product. There is a temperature control also, through which we check the temperature of the uh, fermenter also. There is a thermometer. There is a thermometer and cooling coils and jacket around a fermenter, which which maintain the temperature of the fermenter. During the fermentation process, various reactions take place in the fermenter. Heat is generated. Several reactions is there. Heats are generated. Addition is also may not be addition of the fermentation. Heat is also be there. So overall, we have to maintain the temperature of the fermentation to avoid any such of contamination also, even blast also. Sometimes temperature is very high, heat is excessive, so it may blast the possibilities that the fermenter will blast. But in most cases, this may not be the case. In all such cases, either additional heating or removal of the excess heat would be required. So there will be heating control, cooling control. So we have to maintain the balance of the process, which is described for the following formula. Q is the heat, Q maintain, met, Q egg, gas, excessive evaporation, sense. So all these things can be described with this formula. Where Q met is, if we talk a Q met is the generated by heat generated by the microbial metabolism. Q egg is the mechanical agitation. Q gas is the rate of heat generated by aeration power input. Q X is accumulation of the in the system. Q exhaust exchanger, not exhaust, exchanger, the rate of heat transferred to the surrounding and or heat exchanger, heating or cooling device. Q evap, the rate of heat lost due to the evaporation. Q sen, the rate of sensible enthalpy gained by the flow stream. So Q exchange equal to Q mat plus Q X plus Q gas minus Q X minus Q sen minus Q X. In this equation, Q exchanger provides the estimate of heat that has to be removed by the system. There is another device, sensor you can say, control system is there, is foam control. Foam is produced during a most microbial fermentation. Foaming may occur either due to a medium component, like for example, protein present in the medium, or due to some compound produced by the microorganisms. Proteins present in corn and steep liquor, pharma media, peanut meat, soybean meal, etc. So they may cause the foaming in the media. This protein may denature at the air-broth interface and form a protein film that does not rupture readily. Foaming can cause removal of cells from the media. Such cells will undergo autolysis and release more protein into the media. So there is a foam formation. So they, in turn, there will be a further stabilize the foam. So foaming control device is placed on the top of the fermenter with inlet into fermenter. This device contains a small tank containing anti-foam agent. Foam is generated during, as we already discussed, foam is generated during several processes, during the several reactions, proteins, during agitations also, during shaking also, there are foam formation. So there is a sensor, there is a control point to control the foam. There is a 5 different patterns of foaming are recognized in the fermentation process. Foaming remains at a constant level throughout the fermentation. Initial foaming is due to the medium, but later microbial activity contributes to it. So initially there will be a foaming, but maybe microbial activity contribute more to the formation of the foam. Foaming declines steadily in the initial stage, but remain constant thereafter. The foaming increase after a slight initial fall. In this case, microbial activity is the major cause of foaming. So there, if the autolysis is there, there will be more release of the protein. So microbial activity may cause surplus the foaming formation. The foaming levels increase with the fermentation duration. Such foaming pattern is solely due to microbial activity. A complex foaming patterns that combine features of 2 or more of the above patterns. So what should be the ideal antifoam? We have already discussed in the different lectures about the character property of the antifoam. So antifoam, it should disperse rapidly and act fast on existing foam. It should be used at low concentration, prevent formation of new foam, degrade the uh, other formation of the foam. It should not degrade the microbial activity, microorganisms. It should be non-toxic. It should not interfere with the downstream process. There should no problem in effluent treatment. It should be safe, cheap, and does not affect oxygen transfer. So this is the ideal antifoam which can be used during the fermentation process. There are several types of fermenters are available in the market. So stirred tank fermenter is there. There's a glass small vessels. You can find stirred. There's a for the if you talk about the lab-based fermenter, there is a glass stirred tank fermenter, small vessels, and for the larger scale, there will be a stainless steel, large volume vessels. These are closed systems with a fixed volume and are usually agitated with motor-driven stirred and considerable variation in the design details. Airlift fermenter. An airlift fermenter consists of gas-light buffet riser tube or draught tube connected to a downcomer tube. The aerated medium, both of the riser tube has a lower density, while that in the downflow tube it is a relatively much less aerated and as a consequence has a higher density. The lighter medium in the rise tube flows upward till it reaches the gas disengagement space of the fermenter. So this is the airlift fermenter with extremely placed riser tube. So you can say gas disengagement space, riser tube with baffles, heat exchanger, compressed air spurgers. So we can find direction of the growth flow, downflow. What is the airlift fermenter with inner loop is there. So you can find broth liquid media, culture outlet, bubble breakup device, riser tube, air aroma asperger, sterile media inlet, heat exchanger. So these are the basic diagram of this airlift fermenter with extremely placed riser tube or airlift fermenter with the inner loop. There is another diagram of the airlift fermenter used by Marlow Foods. So you can find the we put the glucose, biotin, and mineral salt pumped in a constant rate. Downcomer tube continuously remove CO2 is removing. Rice tube, vessels, reduction vessels, steam inlet, mycoprotein harvest, culture harvesting port, heat exchanger. So this that is used by Marlow Foods, UK for the production of mycoprotein in a continuous flow. So the fermenter was developed to reduce the production cost by minimizing cooling costs since agitated vessels could be generated additional heat. So the airlift fermenter of 43 MQ volume is used in continuous mode for the production of mycoprotein formed from fusarium and gaminarium, both on wheat starch based medium. So there's another fermenter for the animal cell culture used in such vessels that are both aerated and agitated by air bubbles introduced at the bottom of the vessels. The cells and the medium that lift out of the draft tube move down outside the tube and recirculated. So you can find. So there's a gas outlet, vessels well, cells, draft tube, air bubbles, gas parcel, sparse membrane. So this is the one of the representation of the airlift fermentation for the air cell cultures. There's a tower fermenter also. A tower fermenter has been defined by Greenfields and co-worker as an elongated, non-mechanically stirred fermenter that has an aseptic ratio of at least 6:1 for the tubular section and 10:1 overall. And there is a unidirectional flow of gas through the fermentation. So bubble column, vertical tower, beer fermenter, multi-stage fermenter systems are designed for the tower fermenter. Bubble column tower fermenter, these are simplest type of tower systems. Then they consist of glass or metals tube into which uh, you can say into which air is introduced at the base. Fermenter volumes from 3 to up to 950 have been used, and mainly used for the citric acid production or tetracyclic productions, or for a range of other fermenters based on mycelial fungi. Vertical tower is there. These fermenters were designed for beer production and to maximize yeast biomass yields, having the capacity of 220,000 capacity and capable of producing up to 90,000 beer per day have been installed. Multi-stage fermentation is the column from the body of vessels which is divided into different compartments by placing perforated plates across the fermenter. About 10% of the horizontal areas of plates is perforated. In a variant of this type of fermenter, the substrate is fed onto at the top and overflowed through a downspout to the next section, and the air is supplied for the base. This has been used for continuous culture of E. coli, Saccharomyces cerevisiae, or baker's yeast and activated sludge. So you can find the different different types of, you can say this diagram of the bubble column fermenter fitted with the internal cooling coil. So this is the direction of growth flow, P2, oxygen, air spargers. Okay. P1, P2 indicate location of the pH electrode, O1, O2 extra declare the location of oxygen electrodes. So this is how the bubble cooling fermenter lift. Bubble of fermenter is a bubble column fermenter that is fitted with an internal cooling coil is there. There is a pH control points are there. There is a oxygen flow sensors are there in this bubble of fermenter. These are the simplest type of tower fermenter. They consist of glass or metal tube into which air is introduced at the base. These tower fermenters have been used for citric acid, tetracycline, or as already discussed, the other for fermentation based on mycelial fungi. Thank you.